Hello everyone, welcome back to Dcademy Biology class. Kung ngarang ito wi makada DNA replication do ngarang togre. DNA replication da ay da habitul revision to chiko DNA replication sa. For the DNA replication, I thought the synthesis of DNA. That's right, the synthesis of DNA from DNA to name. Some have it. The synthesis of DNA. So kay gawi DNA replication. Ano DNA ni kaya dagi sung dulo DNA sung dulo ni ko. Single replication. Replication di karamba stage ta tawi hai ta di ko S phase. S phase din jangi ba. Kono cell cycle gi M phase ram ne. M phase ne. Adi interphase S phase ne. Adu di ko G not phase ram lebda. Ada ka do ga S phase sida ka do ga synthesis phase ad. Synthesis phase. Synthesis phase ko. Synthesis phase ito udishi cell cycle di synthesis phase ta. DNA replication never told us. Ada S phase matungda M phase lana. Ada S phase ta DNA replication to aga DNA ki contents ya. Double times lang to neba. Hana DNA layang bapoto DNA ki chang do ki double times lang to ne. Ada DNA replication ji kamay na to we have of the na happy previous class the ID high bani ada may video da yi muda. So that hand written the happy to okay. DNA replication je si DNA strand. Si DNA strand dito ka ba? Sa DNA replication je tao dore hai ta di sa si mapa sida si nga pa sa DNA replication tao dore ai da hai se. Adu ita di sa si nga pa sida yung si nga pa sa DNA replication tao dore. Si ha na laser mi ba? Si nga pa sida si ju suppose si the replication tao de na khalo. Or si one lai de na khalo si one lai ka do dira. Replication tao dore hai ta di si nga pa sida Y shape fork ama form ta di. Y shape fork replication fork ana hai Y shape replication fork. Y shape replication fork ana hai ko. Si Y shape replication fork siya. Kadaw gida masay si nga pa sida to shape. Ada tadi Y shape replication fork sa kadaw DNA replication je. Maga si the parent strand horn. This is a parent strand. Parent strand ke do ba. Helicase habe enzyme na sigai ra ni ko. Sigai ra mai si another new strand was one sima ka do ni. So na kan gisu. Si new strand was one sima ni. Si ni. New strand na color ma use to gi si ai. So new strand ni. Sina new strand. Sina parent strand. Sina parent strand hai. Ada sapo ai na si sumai na yek thora ke ko. Mane to ke si matik siya. Replication da ko yi sumai na yek thora ke. This is a Y-shaped replication fork. Y-shaped replication fork. Sida kay enzyme helicase so sida layer lagi. Kana helicase sabi enzyme lagi. So yaw sida topoisomerase. Ada sida SSB protein layer. Single stranded binding protein. Single stranded binding proteins na kay we destabilize the the helix ko helix ni swaina konjal le bu helix ana DNA je helix model double helix model hai ko je konjal le bu sa mana stabilize to du ne ba sida solang kan da helix ju sida gi siga ka mata da side tension na create ta ro si sida to pu isomerase ni relax da tension ana stabilize de chang ama da thambi de si helical structure je stabilize to ga tham do sina SSD protein is also known as helix stabilizer hanisiko helix stabilizer stabilizing protein hanisiko it is known as helix stabilizing protein and helix have enzymes it unzips the helical structure habadi unzips the word ke sida sumaina ma sina chatlaga sisiga to ni batam tam unzip adi change mana ching tha bai ta ta ja siga to ni sida unzips the helical structure hanji se tele ada mana unzip ta do se kari bone break ta do se kari bone break ta do ja da si adenine ga guanine ga suppose adenine ga guanine ga makta le bone ji kai ta hydrogen bond hai ko aduna mana thubai mana break ta do se hydrogen bond between the nucleotides nucleotides ma ta lai ba hydrogen bond do mana dai thubai do ne breaks it breaks the hydrogen bond between the nucleotides ada sine and 5 prime to 3 prime so 3 prime to 5 prime 3 prime to 5 prime d ada tadi asagi kai la ani ham tam da RNA primer ma darkar oi ko RNA primer ma darkar oi series RNA primer RNA RNA primer sina it is a RNA segment macha mani ko RNA segment macha je RNA primer with the help of the enzyme RNA primer si matung ina RNA primer se sida hapchal ani sida hapchal matung RNA primer se karam ma pa da happe hai ta karam ba DNA si magi DNA parent strand ni karam ba ending the hap kai ta di magi 3 prime end to the hap to name karam ba 3 prime end to the RNA primer se hap to name and then 3 prime ended RNA primer se hapchal matung sida after that the DNA polymerase ya lani DNA dependent DNA polymerase natra DNA dependent DNA polymerase DNA dependent DNA polymerase lani la matung sida RNA primer sida mas mai targa DNA polymerase chana ba nucleotide sing du pura ga lakpi ani chana ba nucleotide sing du sida 8 tawrani loin enter jakhana then a new strand is synthesized ala sida di kayo ni this is a 5 prime end and this is a 3 prime end ko jakhre adagi summa kan side di sida di kamai tawba asagi RNA primer se karamba mapa da happe ba no 3 prime end gi mapa da happe ba no tro aduna RNA primer se karom settle ani si 3 prime pa se lani ko lani aduga sumna kan side sida di kadau ke da si je mate khatani thadali je si mate chi pa chi khatani si makha si yaudri si de sum chakhide de makha gi pa chi yaudri banina asagi sumna kan da template hapaka dusha so kan new strand hapaka dusha only a short template khatani mana phangis only it gives only a short template khatani phangise aduna kadau ge da asagi short strands man hapani sumna kan side di khaidok tri aduna si bokti kadau ge da si bokti sumaina lai ri hai tar sumaina lai ri banina si bokti sumaina khaidok tri sumaina lai ri na phang ni ma pa se karai do no phang ni ma pa se only this portion ji katane ko aduna yeng si after that adu helication na ja sum chakhide helication na ja chakhide ni helication sai sum helicase na si mapasi sila lay mo po ina sumana tawag helicase chakha matung sida adi sumana sangurani naja adi helicase sumana sai dong le sila lay mo po cha ta ja thon ke na sai ji mana strand hydrogen bond break tawre ni topoisomerase lai topoisomerase in helicase topoisomerase adagidi helical bond chakha matung sida kamai tawle hawadi sida kamai na sidi madi dum strand continue da dum synthesize tawre ko this is continue da madi synthesize dum tawre continue da dum synthesize tawre then si na o kadawge da continue da synthesize taw ko da buta asagi RNA primer sha sida synthesize taw ka bane ba si mapa si da si blue se chan sai new strand jo sina sai shung gi mapa se ado sigari batalai mapa se this site ni si site da another primer ma da ka then another primer se sida hapchala right then adagi same case asagi DNA dependent DNA polymerase laga DNA strand ja sana 8 toxalani other like this so na kan ni portion se 3% de cha sambali hai matak samdui other na sigi ka doge da RNA primer se di ngadi remove taw gani ko RNA primer se di remove taw gani RNA primer se remove taw ga matung sida asagi remove taw sa hap samjana ani si samjana ko sida su suppose si chak khaina khalo suppose si DNA fragments ma cha ma le and RNA primer se remove taw gani RNA primer se remove taw sida ang samjana tene ba si DNA strand newly synthesized strand machet form tawega this fragment is known as Okazaki fragments aduna sigi part na madi mangjong na hana DNA synthesized tawga this strand is known as leading strand this is a leading strand ada si taphawe na this is strand is known as lagging strand leg tawbodi thingda huwa da lagging strand and Okazaki fragments is formed in leading strand or lagging strand is formed in lagging strand Okazaki fragments is formed there Okazaki fragments is formed to edu see DNA strands after the RNA primer is removed kadogu the DNA ligase have enzymes la ani DNA ligase have enzymes la ga sai Okazaki fragments sing sa amuk mana samjana do ne samjana do ne right of the newly synthesized strand of DNA si phangye sidagi diketoni newly synthesized strand of DNA this is a old strand new strand old strand new strand and that's DNA synthesized story sini ko nan gi kenadi slide to mo gigs DNA DNA polymerase nucleotide hapcha lagte ba then DNA polymerase on so see in the leading strand lede 5 prime 3 prime whereas the lagging strand so the 5 prime and 3 prime aduga magi template strand so the kai oige 3 prime to 5 prime magi template strand na see template strand na 3 prime to 5 prime aduga 3 prime see template strand leading strand gi template strand kari gi strand do template strand hai ta di leading strand template strand ka direction da chatli leading strand ko na som de thira kani 3 prime to 5 prime hai 3 prime to 5 prime 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 3 prime to leading strand gi template strand. Ada DNA synthesize to obudi adum 5 prime to 3 prime direction na chatli ko. Newly synthesized DNA degi magi direction degi 5 prime to 3 prime na chatle, whereas the template strand gi direction gi template strand di magi parent strand habani ko. Parent strand gi direction di 3 prime to 5 prime. Aduga legging strand gi template ki strand na legging strand ki template ki strand na kari do hai matamda ay wani dudi 5 prime to 3 prime. So the legging strand ki direction. Previous class ta kuna DNA replication bak ta ugre aduga si transcription ni mati ma hauram bani ko. Transcription adi, so what is transcription? Transcription hai ta di DNA degi RNA synthesize to hoto. The synthesis of DNA, the synthesis of RNA from DNA is done as transcription na ko. So transcription da kai changi promoter ma change structural gene ma change terminator ma change. Have DNA, this is the transcription unit of in DNA. So DNA matik ama degi RNA system ko dole. Ala promoter ma dhaka ore structural gene ma dhaka ore then terminator ma dhaka ore. Duga polarity sidana sagi template party ke doi DNA replication is both the strand is synthesized to iko. Both the strand of DNA is animak to ke doi new strands ma animak ta yaugi bodeva same bada. Adabu sagi keno dadi DNA kari transcription dadi ke doi only 3 prime to 5 prime ends ya sina katan template ogani ba. Ado yaudaba synthesize to boda template oidwa pot tuna kai kauge da 5 prime to 3 prime oil. Ada si sina kai kauge da coding strand na kauge kagino da mase RNA ki strand ga chamana do ne except the thymine is replaced by uracil. So thymine is replaced by uracil. Ado na coding strand ga RNA ki strand ga managani. Yang si ko achi makata ga gyan tarakan. Then promoter sidi kai de lai ni ha matamda ase ki sidi promoter ma pade ne ba transcription ho be promoter ma pade di transcription ma di kai da pade is located at the 5 prime end, that is upstream of the structural gene the upstream ma di mamang wai ta lai ha upstream di mamang it is located at the 5 prime end of the structural gene. The presence of promoter also defines the template and coding strand. By switching its position in terminator, the definition of coding and template strands could be reversed. The structural gene has the structural coding for a particular protein is fling, but fling at the site fling at the magi smathangna bahabali. The structural gene the kana thangna promoter na shida lede promoter ga adi terminator gi marakta structural gene lede. So terminator haigana asagi DNA replication or transcription DNA replication transcription jika the we version ma pare. That ends the process of transcription. Asagi transcription ni process ends to do niya. Kada promoter na 5 prime ni ma padale re adugo terminator di kari ma padale hayani it is located at the 3 prime end of the coding strand. Coding strand hasina coding strand ni legi do coding strand on the high coding strand higher than a 5 prime end cycle ni Lochalakwadana 3 prime end cycle later. Adegadi non-coding strand or template strand template strand you can say as it can also be said as non-coding strand non-coding strand the mapada haradi promoter se kari endale hata in with regard to the template strand with regard to haragana promoter se kari shuttle 3 prime end later aduga terminators na kai shuttle 5 prime end later. Then transcription unit and the gene. A gene is the functional unit of inheritance. Or gene is a functional unit of inheritance. The DNA sequence coding for tRNA and RNA molecule also defines a gene. So by the high tRNA DNA की RNA की attachment में दो tRNA transfer RNA or ribosomal RNA. T stands for transfer RNA. R stands for ribosome RNA. Also defines a gene. Cistron it is defined as the segment of DNA coding for a polypeptide. And again, DNA is in a polypeptide chain form talk of the hybrid polypeptide in the amino RNA semacodonia. And the cistron it is defined as the segment of DNA which code for a polypeptide. And again, RNA chain semacodonia DNA. And segment of DNA ma part of the cistron. And then not all the parts of the DNA say RNA same the motive. DNA ki part magi segment magi asang kudimak RNA same only a small segment. Only a small segment. That segment is known as cistron. It was defined as the segment of DNA coding for a polypeptide. And the structural gene present in the transcription unit can be monocistronic polycistronic. Mono only one monocistronic that is consists of one cistron. Cistron DNA code DNA code one cistron it is present in eukaryotes. Whereas polycistronic by hybrid prokaryotes B stands for prokaryotes. Prokaryotes. It is composed of more than one cistron. Cistron amadagi hene yauradi polycistronic. The monocistronic structural genes in eukaryotes have interrupted coding sequence. That is, the genes in them are interrupted or split. Cistron it is one cistron that is one cistron have the DNA. It is a unit of the DNA. It is a part. It is a segment of the DNA which code for a polypeptide. The intron interrupted by the intron parts interrupted. Interrupted habadi. As I guess cistron it codes for polypeptide or RNA semacodonin. RNA semacodosia. Sige, intron RNA semacodonia. This part does not code for polypeptide. This part does not code for polypeptide. Masih polypeptide. Polypeptide siya code tawdi intron capacity. Aduna koida code tawo pami sidi masagi mRNA suloy na makolak pamo dede. Adu si intron siya ng transcription tawo matung sila intron siya sina pre-RNA ko pre-mRNA ko isina intron yawo po si pre-mRNA same lo dede pre-mRNA. Ana same lo badi kang kan lo dino sige intron siya. Intron sila kokakanda. Intron sila kokakanda mature RNA form the structural gene in eukaryotes consists of the following exons. Exons have introns have exons these are the coding sequence or express sequence which appear in mature or process. Mature or RNA da exon yawba. Exon khatang yawdi. Adu intron hai that interrupt the exons and not present in mature or processed RNA. The processed RNA or mature or the RNA sida introns yawdi. Thus, this sequence are called as intervening. Aduna marakta masi intervening tawi habadi mo da marak sida interrupted tawi na intervening ko we have. The intervening sequence had in it. And one of the characteristic feature of eukaryotic DNA is the presence of genes with both exons and introns, which is called split gene. Aduna eukaryotes today, asagi presence of both exons and introns yawani. Introns ka ani magyawani, whereas in prokaryotes there is no exon intron. Ada exon have intron have like team ID aduna pula adum mRNA si adum transcription tawag tadi adum mRNA the form tawag din. So tadi si habitang If the sequence of the coding strand in the transcription unit is written as, transcription at the DNA strand, RNA strand, before this, she didn't have prokaryotic transcription, prokaryotic transcription, in prokaryotes, like bacteria, there is only one enzyme, DNA-dependent RNA polymerase. Replication, DNA-dependent DNA polymerase. This is RNA polymerase that catalyzes the process of transcription. Transcription. The steps involved in the process of transcription are the initiation, elongation, then termination. Initiation is DNA replication or transcription. The first step involves the binding of RNA polymerase to the promoter and also associating with initiation factor sigma. This is a sigma for a short period of time, which initiates the process of transcription. And the initiation is promoter structural gene upstream to the structural gene, we have promoter. Downstream na terminator. This is a structural gene. That is structural gene na code. The promoter the sigma have a factor sigma have a factor then sigma have a factor the RNA polymerase then RNA polymerase na kay porani asagi nucleotide stingdo poraga chain mani polypeptide chain polypeptide chain to aduna asagi initiation je karam template stand do asagi template stand karam stand transcription the template stand is template stand hai tadi code tawduri stand in the template stand she stand de hawi sige stand 3 prime to 5 prime m for 3 prime to 5 prime m in the template stand aduga asagi sida coding strand in 5 prime to 3 prime m coding strand in kay orani wala sagi sigma factor sila DNA RNA polymerase la matung sida kedoga asagi aga complementary of a strand form to RNA. This is a polypeptide chain and this is RNA form to hawi. The enzyme makes up nucleoside triphosphate as substrate in polymerase in a template dependent fission according to the rule of complementary. Say chana ba sam haiga adenine ni mapadana kailan adenine nga kanaga base pair form to diba adenine nga thymine ga and the thymine is replaced by uracil thymine is replaced by uracil aduga adenine cytosine ga guanine ga ma form to sini ko aduga sigi mapasida pagi complementary stand na same to the rule of the complementary stand have the adenine ga uracil ga lani cytosine ga guanine ga the opening of the helix is also facilitated by it. Then elongation have a polymerization polymerization and the sang chain create the step involves the elongation of RNA chain or polymerization. The process in the direction the 5 prime to 3 prime 5 prime to 3 prime carrying the transcription bubble along with the RNA and along with the RNA bubble the cement after initiating the process of transcription, RNA polymerase lodges the factor but continuing to add ribonucleotides to form the RNA polymerization of RNA molecule. So the ribonucleotides RNA polymerase form the Then after that, the termination di nang location di the location di no ay matamda. Sige terminator segment. Sige karam babang pada. Sige terminator segment na lagi jo. Promoter lagi promoter di structural gene na sige RNA polymerase segment atau terminator na pasi da load ni. Also, titration thus terminates the process of transcription. The RNA polymerase associated with the termination factor row have a factor set in a row. How did he? Row have a factor say row factor sila adi. Row factor to stop the transcription process. Sagi row factor sida si pasin la aganda lotion gani. Coupling of transcription and translation process in bacteria occurs because of the following reasons. The mRNA in bacteria, coupling of transcription in bacteria because of the mRNA in bacteria does not require any processing to become active. The process of transcription and translation both takes place in the same end compartment as cytosol and nucleus are not separated. Thus, translation begins much before the mRNA is fully transcribed. And the mRNA translation hagan protein synthesis. This is a protein synthesis. Protein synthesize tawdoni. Ada protein se RNA dai kayong olok tayo na protein na mong siya makulini. Ada sida loy ba na tiba. Sida ka dogi lip tabida niya. Prokaryote sidi RNA dai kayong olani. Protein na dum olani. Habidi translation na dum makata ada ka. Translation na dum saradi. Ada sida isi. The process of transcription and translation both takes place in the same compartment as cytosol and nucleus are not separated. Thus, translation begins must before the mRNA is fully transcribed. So, the mRNA is similar to translation is makata na dum chatharani. Other in eukaryotic transcription the factor of humchangi RNA polymerase 1 habamne, RNA polymerase 2 habamne, RNA polymerase 3 habam. RNA polymerase 1 it transcribes ribosomal RNA minus ribosomal RNA is transcribed. Whereas RNA polymerase 2 is mRNA. That is the heterogeneous nuclear RNA or hygiene RNA short form. Whereas RNA polymerase 3, it is responsible for the transcription of tRNA as a 5SR RNA and snRNA, small nuclear RNA. Siguro siya form tawduni DNA RNA polymerase 3. So yung RNA polymerase 1 la agana RNA form tawni, ribosomal RNA form tawni, RNA polymerase 2 agana snRNA form tawni, 3 agana tRNA, sRNA, then snRNA form tawni. More than the exons and introns are this the exons are interrupted by introns never. Introns they do not code for polypeptide. Many polypeptide code are present in the primary transcript and thus are non-functional. Therefore, the primary transcript RNA undergoes processing, which includes the following. Another sagi, who wanted the two? Sagi, this is a down the part in intron. This is intron. So intron say yaw pochina yaw Muslim took it in intron jihan. Intron took it any intron took it before intron took the other hand first of all we see splicing and high splicing and the it is the process of removal of introns. Splicing at that it is the process of introns took it in here. Then exons are joined in a defined manner, exon gesa. Something then matanga capping habisina no. It involves the addition of methyl guanosine triphosphate. C maton sida C mapa sida kay hapsala kadiba. Methyl guanosine triphosphate hapsala kadiba. Methyl guanosine triphosphate. At the end of the methyl guanosine triphosphate capping to the karam end no 5' end of the hydrogen RNA. Karam end na matamda 5' end of the hydrogen RNA. Then telling in this process, adenylate residues are added to 3' end of the template. Say 3' end of the template sida polyadenyl hanasukobuni polyadenyl siya. adenylation si AA habadi AA habadi karo adenine habana tro adenine jiga do ni si je poly A tail ko poly A tail sa hapchalak tu ni poly A tail sa hapchalak ni then are added at 3' end of the template independent manner after the processing of the HNRNA fully. It is now termed as mRNA. Shidi C hypothetical ko HNRNA has HNRNA te kai wala ani mRNA na fully now termed as mRNA. Aduna si jiga do wi kadogeda si transcription de kai to nucleus manung to le nucleus tagi masika de thadokrani cytoplasm thadokrani out of into the cytoplasm thadokrani cytoplasm thadokra matung sida translation de thokten translation na kai de to ba translation occurs in cytoplasm what is the transcription and the transcription occurs in transcription translation transcription and replication na nucleus tawi aduga translation jina cytoplasm do ana kadogeda translation tawbagi damakta mapan da kasi nucleus ki mapan da thadokrani sis nucleus ki mapan da thadokrani that is out of the nucleus for translation. Genetic code to next topic ta genetic code habadi it can be defined as a sequence of nucleotides before the see to the other way see the solution if the sequence of the coding strand in a translation unit is written as coding strand na C ba coding strand habani ko coding strand na C ta di rather the sequence of the mRNA ki sequence to kai oiduno NCERT ki textbook ta yawo in the transcription if the sequence of the coding strand habani coding strand hai ta di asagi coding strand hai ta di chak manate ba DNA template to chak manani but adu onthok tu si kai dang onthok ani eresa thymine gi mapa sida kari onthok ani ha matanda in the translation if the sequence of the coding strand is since the coding strand is not code for anything it is sequence is the same of the mRNA and then the mRNA the acid is replaced by thymine that's the can change to ba coding strand kari dang change to ni adenine thymine gi mapa da kari replace to ba ni thymine gi mapa da aduna si kamai la ni thymine gi mapa da uracil na simana same ba ni uracil G adenine CDG dum la ni C A C T mapa da U G C A U G C so on and then thymine gi mapa is replaced by U do chamana same sequence and C na mRNA gi sequence hai ada ina question ji da coding strand ta bida ina template strand hara magi template strand hara magi template strand hara bataradi magi sequence kai oina orani adenine ga kana ga orani dudi magi adenine ga thymine ga ha adenine ga thymine ga aduga thymine adenine ga sing midi adenine ga thymine ga ga tabudo adenine ga thymine ga haidani dana adu higher kai oina hani adenine thymine is replaced by adenine aduna kai gumba sina template strand or me ha ta di magi RNA mRNA du kino sa ta kai ojuno da mRNA si pochi kai ojuno da template strand da di kai mai si sequence kai or template strand is 3' to 5' or template strand aduga nana coding nangi strand di mRNA gi strand di kai or ani mRNA gi strand di 5' or ani A sina kai replace to U thymine ga A ga guanine ga cytosine ga guanine ga adenine ga thymine di U A T ga thymine ga adenine ga guanine ga cytosine ga A sina kai replace it is A sina thymine di kai na olani U minus so on and after them template strand or gonna adi complementary strand on to over have the adenine or gonna thymine di era you can RNA di uracil ni right should ꯃꯊꯪꯗ genetic code ꯑꯁꯤ ꯇꯧꯔꯁꯤ genetic code ꯍꯥꯏꯕꯗꯤ genetic code ꯍꯥꯏꯇꯥꯔꯗꯤ ꯉꯁꯥꯏꯒꯤ polypeptide can be nucleotides in DNA and RNA which determines the sequence of amino acids in a polypeptide chain or a set of certain rules according to which the information encoded within genetic material is translated into amino acids and proteins. genetic code ꯑꯩꯈꯣꯏꯅ code language ꯀ ꯍꯥꯏꯕꯗꯣ code language ꯍꯥꯏꯔꯗꯤ ꯈꯪꯍꯟꯗꯕꯤꯗ ꯅꯪ ꯋꯥꯔꯤ ꯁꯥꯟꯅꯕꯗꯨꯅꯦꯕ ꯑꯗꯣ code ꯍꯥꯏꯇꯥꯔꯗꯤ ꯅꯪ ꯀꯩꯅꯣꯝꯃ ꯃꯤꯉꯣꯟꯗ ꯍꯥꯏ ꯅꯠꯇ꯭ꯔꯣ ꯀꯩꯅꯣꯝ inform ꯇꯧꯔꯤꯅꯦꯕ ꯑꯗ ꯀꯩꯗꯧꯒꯦꯗ genetic code ꯍꯥꯏꯇꯥꯔꯗꯤ ꯉꯁꯥꯏ sequence of DNA ꯁꯦ sequence of DNA ꯁꯦ or DNA ꯑꯣꯏꯕ ꯌꯥꯏ or ꯅꯠꯇ꯭ꯔꯒ RNA ꯑꯣꯏꯕ ꯌꯥꯏ ꯁꯤꯁꯤꯅ ꯀꯔꯤ code ꯇꯧꯔꯛꯀꯗꯣꯏꯅꯣ ꯍꯥꯏ ꯃꯇꯝꯗ geneticꯀꯔꯤ amino acid ꯑꯃ code ꯇꯧꯔꯛꯇꯣꯏꯅꯦ amino acid ꯇꯧꯔꯛꯇꯣꯏꯅꯦ sequence of a polypeptide chain or a state of rules according to which the information encoded within the genetic material is translated into amino acid. ꯑꯗꯨꯒ ꯉꯁꯥꯏꯒꯤ genetic code ꯀꯩꯅ ꯑꯣꯟꯈꯤꯗꯣꯏꯅꯣ ꯍꯥꯏꯕ ꯃꯇꯝꯗ ꯉꯁꯥꯏꯒꯤ amino acid code ꯇꯧꯈꯤꯗꯣꯏꯅꯦꯕ amino acid code ꯇꯧꯔ ꯃꯇꯨꯡꯗꯗꯤ ꯉꯁꯥꯏꯒꯤ gene expression ꯖꯤꯟꯁꯦ ꯃꯥꯒꯤ ꯃꯑꯣꯡ ꯃꯇꯧꯁꯤꯒ amino acid ꯁꯤ ꯑꯣꯏꯇꯥꯔꯗꯤ ꯃꯥꯒꯤ ꯃꯑꯣꯡ ꯃꯇꯧꯗꯣ ꯀꯩ ꯑꯣꯏꯔꯅꯤ ꯃꯥꯒꯤ character ꯑꯣꯜꯂꯛꯇꯣꯏꯅꯦꯕ ꯑꯗ DNA or RNA ꯁꯤꯅ code ꯇꯧꯕ ꯍꯥꯏꯕꯗꯤ ꯀꯩꯗꯣ ꯑꯣꯜꯂꯛꯀꯗꯣꯏꯅꯣ ꯍꯥꯏꯕ ꯃꯇꯝꯗ amino acids ꯍꯥꯏꯕꯗꯤ or protein ꯑꯣꯜꯂꯒ ꯉꯁꯥꯏꯒꯤ ꯖꯤꯟꯁꯦ express ꯇꯧꯕꯁꯤꯕꯨ ꯑꯩꯈꯣꯏꯅ genetic code ꯑꯅ 하이드님. So it can be defined as the sequence of nucleotides in DNA and RNA which determines the sequence of amino acids in a polypeptide chain or a set of certain rules according to which the information encoded within genetic material, DNA or mRNA sequence is translated into amino acids in a protein. ꯑꯗ ꯃꯈꯜ ꯁꯥꯏꯟꯇꯤꯁ ꯃꯌꯥꯝꯅ ꯖꯣꯔꯖ ꯒꯦꯃꯣ ꯍꯔꯒꯣꯕꯤꯟ ꯈꯣꯔꯥꯅꯥ ꯃꯥꯔꯁꯦꯜ ꯅꯤꯔꯟꯕꯔꯒ ꯁ꯭ꯇꯦꯚꯦꯔꯣ ꯑꯣꯀꯨꯋꯥꯟ ꯁꯤꯒꯨꯝꯕ ꯁꯥꯏꯟꯇꯤꯁ ꯃꯌꯥꯝꯁꯤꯅ ꯃꯈꯜ ꯃꯌꯥꯝ ꯄꯨꯊꯣꯛꯅꯤꯀꯣ ꯃꯥꯅ ꯖꯣꯔꯖ ꯒꯦꯃꯣꯁꯤꯅ ꯐꯤꯖꯤꯛꯁꯁꯦ 1954ꯗ ꯃꯥꯅ amino acid 20ꯁꯦ code ꯇꯧꯕꯁꯦ ꯀꯩꯗꯧꯒꯦꯗ 3 nucleotideꯅ ꯁꯥꯅꯤꯅ ꯃꯥꯅ ꯍꯥꯏꯔꯝꯃꯦ amino acid 20ꯁꯦ ꯀꯩꯗꯧꯕ 3 nucleotideꯁꯅ ꯁꯥꯅꯤꯅꯀꯣ 3 nucleotideꯁꯅ amino acid ꯑꯃ code ꯇꯧꯗꯣꯏꯕ ꯃꯥꯅ ꯍꯥꯏꯔꯝꯃꯦ अदा कलोगे हरगोबिन खोराना सिन it developed a chemical method for the synthesis of RNA molecule with defined combinations of bases and a base bases atadi adenine na bi guanine na bi cytosine na bi sig adenine guanine siga pullaga kaloge da karamba pochina karamba amino acid olago the bases karamba pullaga karamba amino acid olago the sina kanana karamba hurgobind khorana he developed a chemical method for the synthesis of RNA molecule karamba bases na RNA molecule sarakadino ana hayab then Marshall Nirenberg sina kai haba he gave a cell-free system to provide synthesis that help in deciphering the code jagi code se mo protein synthesis ki haidi code to mana pibirame he gave a free cell system for protein synthesis that help in deciphering the code the Siburo Okwa he synthesized an enzyme named polynucleotide phosphorylase that help in polymerizing RNA with defined sequence in a template independent manner enzymatic synthesis of the minor enzymes in the Kedoba Sagi RNA Shumbugi Metadaman Hayam. Other years Shinigua of the code hide genetic code hide. Genetic code is phenylalanine, leucine, serine, Simiamje. These are the names of the amino acids. Amino acids are common class 11 and amino acids. So far, there are 20 amino acids. 20 amino acids. 20 amino acids. Yeah. That'll get my short form. This is phenyl. This is a short name for phenylalanine. Phenylalanine. The serine. Serine. So I have different names like that. Like had a short form like that. Because get the Marie level sorry, and me to level sorry. Have the you see sina kari see the code sina kari amino acid. All the code kari amino acid ne mana code taisha. Phenylalanine have amino acid ne mana code taisha. UUA sina nucleotide code 3 nucleotide base have the 3 to the 1 2 3 1 2 3 that's all. The 3 nucleotides sina it codes for a amino acid among code to the C UA C sina ani si code ani sina kadawgi sin nucleotide code ani sina nu sina ba entire form to adagi UCU C sina serine ana mari le basiawre adi ani khatang le basiawre mari le basiawre ani khatang smai Chine. Ako stop codon sila ka stop codon chikon hai. The major features of genetic code are as follows. mRNA na AGU. mRNA sina ka ba? AGU ada tadi si ahong hado sini ko tarong hong na. Pero tadi ahong sina ka dawi. Kay form tar si codon na mong ko ahong hado tadi si ha the mRNA or ba siya. This is a mRNA na cha. mRNA is a polynucleotide chain. There's a polynucleotide chain ni mRNA di kay olage codon na di kay ni ba ahong pair ahong shebdo. Sige pair ahong jina 3 nucleotides ahong jina amino acid among code taw ni ba. Amino acid among code to suppose sige kay hay no. AUG matamda kari AUG matamda kari amino acid mana code taw AUG matamda methionine haba amino acid ni mana code taw The codon is triplet hai. Codon nama hai ta di codon is so what is codon? Codon is a triplet of nucleotides which code for an amino acid amagi code taw na ba? Amino acid am all codon ni. And a codon is a triplet of nucleotides which code for an amino acid ama sima code taw du codon and hai. The codon is triplet. 61 codons out of 64 codons like what? So 61 codons na kada 20 amino acid code ne. Sana siguro 20 sina may methionine serine ni namo ni leucine ni namo ni sina. Ada therefore these codons act as terminating or stop codon. Lemo libo 64 imanung sida 60 to 63 sidi. Adi amino acid amatagi code taw diba. Amino acid amatagi code taw din. Sige code sige akong lemo libo akong siya. Sige ko stop codon na ko. Stop codon hai ta di translation dito yari translation taw no na ana stop codon na signal ang plate ni. Ana stop codon hai di translation taw mun hai de. Ko adani. Habadi sida kay gumba suppose sige UAA sida kay stop codon ni sequence siya. Stop codon stop codon ni sequence niyo. Stop codon ni UAA stop UAG stop UGA si ahong si stop codon ni nucleotide codon ahong ne ba ko codon ni stop codon gi CUA oran ba yai UAG oran ba yai UGA oran ba yai ko or ni ko si ahong ji no UAA oran ma di yare translation to lepo na haide UAG oran masu UGA oran masu Ana sige mapasida kay gumba UAA ne oram masine khallo ata si amino acid ang code taw re kacha masi kay do this is a stop codon amino acid ang code taw re si stop codon oire ata si translation ja translation to khani translation di kay do RNA dagi protein sumosina kay translation ana amino acid dagi protein ola ka do ni ko ada polypeptide habadi kan polypeptide ada chain of amino acid habadi chain of amino acid is not polypeptide ada si amino acid ki chain ama sima kan da protein ola ka do ni ada mRNA dagi amino acid ole si yani ni ko ada code che hom sina sana ga methionine be ai serine be ai si code hom sina mana hai matung ina mana code to matung ina amino acid ma pore adi adu kai gumba UAA ota adi translation lo chalo I name UAA or UGA the or share si hom je UAG over here, UGA over here. UAZ over here, UGA over here. Genetic code is degenerate, which means that all amino acids except methionine, AUZ, and tryptophan, UZZ are coded more than one codon. Ano? Hoy, ani sidi codon amakatang ni methionine is codon kya lay amakatang. AUZ katang lay. Tryptophan is codon kya lay amakatang lay. Atay sidi ani ubuyawi mari ubusyawi si. So for example, ZCU, GCC, and GCA, GCZ, C code se kari alanin. Alanin idi codon kya lay ya? Mari leba. Whereas methionine di my codon sidi only one katani, AUG katani, tryptophan na UGC. Aduna kadogida codon si amade hena lay bisu yawi haba. So more than, for example, alanin idi codon mari lay. Yung to sige chart sida. Sige chart proline gisu codon mari lay. Lysine gisu codon mari lay. Serine gisu codon mari lay. Adagidi methionine gidi amakatang lay. This is mo sidi codon ang katang lay ba AUG, tryptophan si codon ang katang lay. Tryptophan sila to UGG utandi tryptophan ni mana ko to siya. Adagi serine di UC or siya UCC or siya UCA or siya UCG or siya mana codon siya more than one or more than one lay bisu yawi haba ko. Aduni is. The genetic code is nearly universal. This means a particular codon codes for the same amino acid in all the organisms except in mitochondrial codons and some protozoans. For example, from bacterium to human, you could code for hadik my code to the chain scheme to have the universal one and chummagalay. Sometimes you na code of this if alanin, not sitting of it kanang sitting at the chain so but like the kayo dung ole diba. Finalanin yung code of the name. Then AUG is na dual functions ka. May function naman na methionine code tawre. Ama na mas initiator codon. Habdi initiator din sa translation si hawo na sin initiator codon ko si yawi. UAA, UAG sina stop codon di masing kala 3 codon ni stop codon di. The genetic code is non-ambiguous and specific, which means a specific codon and will only code for a particular amino acid particular. Tudang code ni ito po kanang code. So this is known as non-ambiguous. Okay, nga sige kung na transcription ni matang po na kang de, the genetic code to buhaw where the mga tungta ko that translation genetic code kang di transcription na ani, then structure of the RNA la ani. Okay, have a great day. Stay tuned. Bye.